All right, welcome back, everyone. So in the last lecture, we went over how solutes can cross through our semi-permeable membrane through simple diffusion or facilitated diffusion with the help of some sort of integral protein or transport protein. And we ended talking about active transport, where we can have solutes go against their concentration gradients with the help of some energy through the form of ATP. So we're going to continue looking at one more part of active transport, which has to do with the idea of bringing in bulk items into and out of the cell. Okay, so bulk transport across the plasma membrane occurs through two different processes. And if you remember from the last lecture, the Amoeba Sisters video kind of went over them. So we have exocytosis and endocytosis. So in exocytosis, think exiting the cell. So that's when the cell is getting rid of, a certain solute. Whereas endocytosis, coming in. Okay, so that's the difference between the two. When we have this exo and endocytosis take place, we're going to basically have let's say if we're talking about endocytosis, the larger substance, it could be some sort of macromolecule, it could be, food particle, whatever it is, it's large and it can't just simply be, fit through a transport protein or anything like that. So instead, it's going to cross the membrane by basically having the membrane pinch off around it and encapsulate it in a transport vesicle. And in order for this to happen, we do need some energy. So this exocytosis and endocytosis are forms of active transport. Okay, so let's take a closer look at this process with a nice visual. Okay, so here you can see we have an example of exocytosis and endocytosis. So in exocytosis, we got substances being exited from the cell. So you can see how maybe these are the products of the Golgi apparatus. Okay, so whatever, glycoprotein or, glycolipid, whatever was formed, is now encapsulated in a double membrane, a phospholipid bilayer, in a transport vesicle. And you can see how basically that transport vesicle is going to fuse with the plasma membrane. So you can see how that lipid bilayer, since it's fluid, is going to kind of rearrange itself and become basically the plasma membrane is expanding at this time, and that is going to allow the substances inside that transport vesicle to leave the cell. And then we have the opposite occur with endocytosis, so that those substances are fusing with the plasma membrane, and then the plasma membrane is pinching around it to bring it into the cell. Okay, so let's look at a video to kind of explain this. Here we have an example.
Endocytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure.
Okay, so you saw how the plasma membrane is kind of expanding when we have exocytosis, and it's kind of shrinking as we have endocytosis take place. Okay, so here are some more examples. So we have a couple examples of endocytosis here. So in endocytosis, remember, we're bringing in a large molecule into the cell. And so if it's a case of some sort of like food or a larger particle, the form of endocytosis we're talking about is phagocytosis. So phago, think like eating. And so we're going to bring that inside. We saw in the Amoeba Sisters video how these unicellular protists, these amoebas, they can actually do that. So they basically extend their pseudopods, because they're a single-celled organism, until they basically envelop that food item and bring it into the cell's interior. So that's called phagocytosis. In pinocytosis, it's a little bit different in the sense that we're going to have a special coated protein that's going to allow certain solutes to enter the cell that are not necessarily large. Okay, so it's basically bringing in something smaller, but through the same mechanism where we're creating this transport vesicle instead of it just going through a protein. And then in receptor-mediated endocytosis, we have these little receptors on the plasma membrane. And so they're going to allow the docking of certain solutes to them. And then once they are fully docked in place, then we have that cell will go ahead and pinch off the plasma membrane surrounding it. So this receptor-mediated endocytosis is very specific. So only a certain type of molecule can bind to those receptor sites in order for that molecule to enter the cell. Okay. So here's a checkpoint question for you. As a cell grows, its plasma membrane expands. So does this involve endocytosis or exocytosis? So when the plasma membrane is growing, is that from endo or exocytosis? So if you chose exocytosis, I would agree with you. Okay, so that's where the interior of the cell, those transport vesicles fuse with the plasma membrane, so that now it's expanding. Okay, endocytosis, the transport vesicles are coming off, are being created from the plasma membrane, so that's when the plasma membrane shrinks a little. Okay, so let's go into a little bit more detail of endocytosis with a human example. So there's different conditions with relation to how our body stores cholesterol, and there is a genetic disease called hypercholesterolemia, and in this particular disease, the cells are missing a certain receptor site on the plasma membrane. It's called the L receptor site. And this receptor site is basically going to allow for cholesterol to bind to it, or what we call the low density lipoproteins. Okay, so the LDLs. You might know them as the healthy part. So you want or excuse me, the unhealthy part. So like in your cholesterol readings, you always want high HDLs and low LDLs. So people with this disease, they don't have the receptor sites that where these LDLs attach. And that means the LDLs are just floating there in their blood vessels, and they can start accumulating in the bloodstream and basically cause something called arteriosclerosis, or the scarring of the blood vessels, and that could lead to a heart attack. So hopefully you don't have this particular disease, because again, it would be, you would have these little receptor sites on your plasma membranes, and so the LDL is able to attach to those receptor sites, allowing your cell to process that cholesterol, okay, and bring it inside through endocytosis. So here is an example of receptor-mediated endocytosis. So normal cells would have those receptor sites allowing the cell to bring in the cholesterol and process it. Where does it get processed again? The smoothie arc? Okay, but if you don't have those receptor sites because you have this genetic condition, hypercholesterolemia, it's going to make it harder for your body to process those LDLs. So they end up gunking up your plumbing, basically, your blood vessels. So some people have, we'll get into genetics later in the term, but some people, they have a mild form of the disease because they only carry one allele for it. And then some, they have the severe form, which means that they don't have any of those receptor sites. So they are very prone to heart attacks and things like that. Okay, so that was an example of receptor-mediated endocytosis in a real situation for humans. Okay, so we've talked about different forms of diffusion. We ended with active transport with the bulk products like our endo and exocytosis. So now we're going to focus on basically the outside of the cell and how the cells communicate with each other. So we're going to take a look at how the plasma membrane is involved with chemical signaling. So as a multicellular organism like yourself, you need your cells to be able to communicate with each other so that they function in unison to coordinate whatever activity that they're doing. And so this is in order for the cells to communicate, they're going to have to use their plasma membranes because that's what typically is coming in contact with each other. And this is not just for multicellular organisms. Our unicellular organisms also need to communicate with other unicellular organisms. So cell signaling is very essential to basically all forms of life. So we're going to start out with an Amoeba Sisters video to kind of introduce us to this because it can be a pretty complicated topic.
A question for you. Have you ever played the game Telephone? You know, the game where someone whispers something to someone, and then that person whispers it to someone else, and then that person whispers it to someone else. And by the time you get through everyone playing, the original message is all messed up. I used to kind of dread that game. Most people seem to like it, but somehow whenever it reached me, the message was always really messed up, so I felt like I was spreading nonsense. Well, either that or it was me all along that misheard it. Anyway, the game is all about communication and how things spread. Communication is incredibly important, not just for us, but for the things that we are made up of, like our cells. Cells make up all living things. And while they don't talk in the way that you and I do, it's important for their messages, their signaling, to be transmitted and received appropriately. Multicellular organisms need their many cells to be able to work together to carry out functions. Just consider all the cells working together in one of your organs, like your heart, for example. We're going to be talking a lot about receptors. A receptor is a molecule, such as a protein, where a signal molecule can bind. One place you can find receptors is on the surface of a cell membrane. When a signal molecule binds the receptor, amazing things can happen. The receptor might start activating another molecule for an action to happen. The receptor often changes its shape slightly in the process. More about that later. So, receptor signal molecules. The signal molecule can be considered a ligand, a fancy term which basically means that it's the smaller molecule that binds to a typically larger molecule. Signal molecules can be a variety of things, such as gas molecules or hydrophobic biomolecules like lipids or hydrophilic biomolecules like some kinds of proteins. But ligands are generally smaller than the receptors they bind. Ligands and receptors can have a very specific fit as well. Let's talk about the general sequence of cell signaling. First, reception. Typically, a signal molecule binds a receptor. Second, transduction. The receptor gets activated by this binding. This often means the receptor will change its shape. It could even involve a whole series of molecules changing their conformation in something called a signal transduction pathway. This can amplify the original signal. Third, response. There's some kind of response that is going to happen. A portion of DNA that is found in the nucleus getting transcribed, for example, that's a type of response. Now, cell signaling can involve intracellular signaling, which occurs within the cell itself, and intercellular signaling, where a cell communicates with another cell. In many cases, signaling involves both, signaling between cells and then also signaling within cells. Now, when we're talking about signals traveling from one cell to another, distance matters. Some cells are close and have direct contact. In the case of these two animal cells, they are gap junction close. Gap junctions in these animal cells, or plasmodesmata in plant cells, are connections between two close together cells that allow ions or other small molecules to pass, and by doing so, they don't have to pass across the cell membrane. Paracrine signaling allows a cell to target another cell by a signal molecule that may diffuse between them. These cells are still close but need not be connected. The ligands in paracrine signaling tend to be rapidly absorbed and rapidly degraded. The ligands are typically not traveling far, as this is local signaling. Synaptic signaling, which specifically involves neurotransmitters in a synapse, is another example of this local signaling. And what about long distance? Endocrine signaling can allow a cell to communicate with a target cell from far away. Signals may be carried in the bloodstream. Hormones released by certain types of endocrine cells are a great example. We also want to point out that a cell could just signal itself. For example, in autocrine signaling, a cell could secrete a certain type of molecule which then binds to its own receptor and causes a response. A cell releasing its own growth factor could be an example of this. Let's just give a few signaling examples so we can get a basic understanding of the vocab and what this can look like. Let's say we have a steroid hormone that travels through the cell's semi-permeable membrane. Remember, that would mean the steroid hormone is our signal molecule, our ligand. Once inside the cell, it binds to a protein receptor within the cell. Now, the protein receptor is active. The protein receptor travels into the nucleus where the cell's DNA is found. This protein receptor binds to DNA and is involved in getting transcription of a certain gene going, which eventually can be used to produce a specific protein. This is an example of signaling inside the cell. Simplified a bit. Now, in the example, the receptor was inside the cell, a cytoplasmic receptor. But it doesn't have to be. Receptors are frequently part of the cell's membrane surface. If a receptor is sitting outside of the cell membrane surface, then the ligand doesn't have to come in. In fact, there could be properties of the ligand that may not allow it to pass through the membrane. The ligand could be hydrophilic, which would make it hard to pass through. Let's mention an example of a cell surface receptor type, a ligand-gated ion channel. Ions normally don't go unassisted through the cell membrane. They are charged after all. See our cell transport video. But a ligand-gated ion channel gives them a way through a channel. But it is controlled. In this example, the channel is closed. But here comes a signal molecule, a ligand. It binds the receptor, which is the channel protein in this case. Now the channel protein responds by opening. The ions, not the ligand, go through. Once ions get through, the concentration of ions increases in the cell. And you may wonder, okay, so now there are ions in the cell. Why does that matter? Well, the increasing ion concentration can trigger a cellular response. After reaching a certain concentration and stimulating a cell response, the ligand can leave its binding site from the receptor and the channel can close. Ligand-gated ion channels can be used by neurons. A neurotransmitter may be the ligand for the channel to open. This could happen at a synapse. But just be aware that not all ion channels are ligand-gated ion channels. Ion channels can be gated by other things. A voltage-gated ion channel, for example, depends on electrical membrane potential, not a ligand. By the way, voltage-gated ion channels are used by neurons too. Check out action potential in neurons to learn more. There are other types of cell membrane surface receptors that we don't have time to go into in this video. G-protein linked receptors and enzyme-linked receptors are two other types that we encourage you to explore. So in summary, why do we care about the cell signaling thing? Well, realize that your body processes that keep you alive rely on your cell's ability of cell signaling. From the regulation of your heartbeat to the hormone signals traveling long distances in your body to the way neurons in your brain communicate, your multicellular self needs cell signaling. But there are many disorders where cell signaling does not work as it should. And so understanding all of the complexities of cell signaling is critical in order to find ways to treat them. Cancer is an example of a disease that can involve body cells with problems in cell signaling. We mentioned autocrine signaling. A cancer cell could have a problem where it produces too much of its own growth factor, causing excessive cellular division. Cancer cells can also have many other cell signaling difficulties where they do not function like normal healthy cells. Or another example, there are also pathogens, such as viruses or bacteria, that can take advantage of cell signaling. Consider the virus HIV, which targets helper T cells. Helper T cells are important immune cells in your body, and they have something called a CD4 receptor on their surface. That receptor is so important so that helper T cells can communicate with other immune cells. But as mentioned in our viruses video, HIV targets that CD4 receptor. It is because of the virus binding to that receptor on helper T cells that the virus can attach and infect the cell in the first place. a fact that continues to be researched for treatment options. The understanding of the details of cell signaling continues to expand. Well, that's it for the Neva sisters, and remind you to stay curious.
All right, so that was a good introduction into cell signaling. And I like that example at the end with the HIV and the receptors, because that's very similar to how COVID works, where it binds to a protein receptor on our cells and allows it to invade the cell and replicate. So let's talk about how cells can undergo different forms of cell signaling. So from that video, it described how cells can communicate with each other through direct contact. So that was where we have those gap junctions, where two cells are so close that they have those little tunnels, those little gaps that allow for solutes or whatever to be transferred from one cell to another directly. So those again are called gap junctions in our animal cells, and in plant cells it's called plasmodesmata. So that allows for substances to go from one cell to another. So that's an example of local distance signaling where we have direct contact. We can also have that video described a type of local signal where there's not direct contact, where you saw how basically that the cell is secreting its some sort of signaling molecule that then gets received by the nearby cell's receptor sites. So that again is going to be a local form of signaling, but not in direct contact with each other, just one of them secreting that signal protein and the other cell is receiving that on its receptor sites. And that there's all sorts of different types of these signaling, local signaling. So again, that's basically, we can have something called peregrine signaling. So in peregrine signaling, we have, you can see the secretion of some sort of chemical or some sort of substance, and then that's going to reach a nearby target cell. And sometimes those signals could stimulate the growth of those target cells, or it can tell it it's time to divide it, because there's something going on with this one. So there's a number of things that this can be used for in this local signaling. Okay. Here's in a more specific type of local signaling. So this is called synaptic signaling. So this is where we have basically two neurons. There's always this gap in between the neurons called a synapse. And that little gap is so that there's no direct contact between the two neurons. But the neurons, they need to be able to continue passing that signal on throughout your system. So basically you have the axon, the tail-like part of that neuron is going to release something called a neurotransmitter, and that could be that's a cell signal. And that cell signal, that neurotransmitter, is going to be received by the dendrite end of the target synapse, or it could be another cell. So these little receptor sites are going to allow for those neurotransmitters to bind to it, and that's going to cause a response in the target cell or maybe another nerve cell. Okay, so this is an example of synaptic signaling, because we're using a neuron, and that's going to be some sort of electrical signal. Okay. So that's a little different from the peregrine signaling, which is just between two cells and it's not electrical. Okay. And then finally, the video talked about long distance signaling. So an example of long distance signaling is our hormones. Basically, if you think about, for example, our pituitary gland, so that's this gland that's in the middle of your brain, and that's going to secrete all sorts of different hormones. Okay, so it might secrete a hormone that could trigger a response in your hypothalamus, another endocrine gland. And then that hypothalamus can then release another hormone that can target another part of your body, such as your ovaries or testes. So basically, we're sending out these chemical signals. So these are a form of chemical signals, a hormone that can then travel through the bloodstream to the target organ. Okay, so like hypothalamus in the brain, all the way down, to send a message to the ovaries or the testes to start egg production or sperm production, for example. Okay, so it's going to be traveling through the bloodstream, that chemical signal, a long distance. So hormones are those, you can think of chemical messengers or chemical signals that travel far distances. And then what is this cell signaling all about? So in the video, it talked about the pathway. So we start, basically they had it, the signal cell, or excuse me, the signal molecule, transduction, and response. Ours book has a little slightly different pathway. It's described as reception, transduction, and then response. So there's not doesn't start with a signal, but it basically is a signal. It's just reception of the signal. This process, this signal transduction pathway was first discovered by Earl W. Sutherland, and it was in response to how the hormone epinephrine is reacted upon a cell. And so the first stage is that reception. So remember, we have some sort of signaling molecule, which we call a ligand, is going to bind to a receptor protein on the cell membrane, okay? And that's going to cause a response. So that response is that transduction, so conversion of the signal to some sort of cellular response. So it might trigger a response within the cell. Okay, and you can see that the transduction phase could be, you know, a certain pathway where we have multiple things going on until finally we get the response to the cellular activity that the signal initiated. Okay, so what the cell was supposed to do. So with epinephrine, you can see how we have, you know, that signaling molecule, which in this case would be the hormone epinephrine, and that's going to cause transduction. So it's going to cause a series of reactions to occur inside the cell, such as the activation of glycogen phosphorylase. Okay, so that will be the end response. So let's watch an example of this signaling of the different stages.
Cells communicate with one another by means of chemical signals. For the receiving cell, there are 3 stages in the signaling process: reception, transduction, and cell response. The cell targeted by a particular signal has a receptor molecule complementary to the signal molecule or ligand. The ligand fits like a key in a lock and triggers a change in the receptor molecule. Signal transduction converts the change in the receptor to a form that can bring about a cellular response. This might involve a series of steps, a signal transduction pathway that alters and amplifies the change. In the 3rd stage of cell signaling, the transduction process brings about a cellular response. This can be any of many different cellular activities, such as activation of a certain enzyme, rearrangement of the cytoskeleton, or activation of specific genes.
Okay, so that was a good overview of cell signaling with the 3, the order of the processes. So we always start with the receptor or the reception stage, where that ligand binds to the protein on the plasma membrane, and then that's going to signal the transduction pathway. Okay, so that's going to relay the message until we end up with a response, the cell's response to it. Okay, So let's go through the terminology, make sure we're understanding everything. So the receptor, again, is going to be the protein that's embedded in the plasma membrane. And that receptor has a very specific reception site where the signaling molecule, which is known as the ligand, is going to attach to. So it's a very, very specific receptor site. So only that particular signaling molecule can attach to it, or that specific ligand can bind to it. And what generally happens is that ligand is going to trigger a change in the shape of the receptor. So that shape can cause a change in the plasma membrane in that protein. So many receptors are activated by that change of shape. So it tells the cell membrane, what to do. So most signal receptors we talk about are basically proteins. Not all of them, but basically for the purposes of this class, most of them are going to be protein receptor sites. So let's take a closer look at those receptor sites. So receptors in the plasma membrane, most of them are going to be water-soluble signal molecules that bind to that specific protein. So that makes sense because outside the cell, it's going to be that extracellular matrix, which is basically, a bunch of water with substances inside. And so the ligand, the signal molecule, will be basically water soluble, and it's going to bind to the receptor protein. And then we're going to talk about two specific types of membrane receptors. So in the video, it didn't really allude to this G protein coupled receptor. So we'll talk about that one specifically. And then we're also going to talk about the ligand-gated ion channel, which the Amoeba Sisters video did talk about at the end. So we'll get into both of them in this class. So we're going to start with this G protein coupled receptor. Okay, so we abbreviate instead of saying G protein coupled receptor over and over again, we can just refer to it as the GPCRs, and that's basically where we have a specific type of receptor site that works with something called a G protein. So if we take a look at our activated G protein coupled receptors, so this is some sort of protein molecule that's embedded in the plasma membrane. And on the inside of the plasma membrane, we have something called a G protein, okay? And then on that GPCR, we have basically the little signaling molecule, that ligand that is specifically bound to that receptor site. So these GPCRs, the pathways are very diverse in function. Okay, so they can elicit all sorts of different reactions. So in this case, you can see how this particular GPCR, once it receives the ligand, it's going to activate a G protein. And that G protein is going to then move inside the cell membrane to activate some sort of enzyme right here to initiate a cellular response. So there's a diversity of functions that can result from this. Okay. And then in a ligand-gated ion channel, that's the one that was introduced in the Amoeba Sisters video, we have the ligand is going to basically, when it's bound to the ion channel, that protein, it's going to open the gate. Okay, so that protein will change its shape to allow specific ions to enter the cell. Okay, so those very important with the nervous system. We talked about how the sodium potassium pumps, so those are ligand-gated ion channels. So once a certain ligand attaches, it's going to allow that ion channel to open up and allow certain things to diffuse through. Okay, and that can trigger an electric signal. So let's take a closer look at that. So here we have our ligand-gated ion channel. So when it's closed, that means that there is no ligand or signaling molecule attached to it. Once there is a signaling molecule or ligand attached to it, it's going to trigger a change in that ion channel to open up. And that's going to allow those ions to travel through and elicit some sort of response. Once the response has taken place, then the signaling molecule will leave, and that causes the closing of that ion channel. All right. So let's look at an example of basically how we have the reception, the transduction, and then the response with an example. So this is an example using a steroid hormone. And in this case, that hormone is going to, since it's a lipid-based hormone, it's going to pass directly through the plasma membrane. So there's no, in this case, there's no protein channel or anything like that. And it's going to bind to a receptor protein, not in the plasma membrane, but within that particular cell. So that's where reception is going to take place. So the hormone is going to be the ligand, and the receptor protein is within the cell. And that's receptor protein is going to activate the receptor, the response. So that's going to basically cause this hormone receptor complex that has the hormone and the receptor protein to enter the nucleus of the cell where the DNA is. And that's going to trigger the DNA to basically send a signal to, in the form of messenger RNA. It's going to activate a gene, a specific gene, to trigger a protein to be made within the cytoplasm. So we have our reception taking place inside the cell, so not on the cell surface. We see the transduction where the pathway is taking place, where the cell signal is going to be received and utilized until we end up with a response. A new protein is being formed. Here's another example. This is called a phosphorylation cascade. And in this case, we see that the transduction pathway is actually pretty complicated. So we'll start with the reception first. Okay, so here this is a receptor protein embedded in the cell membrane. Here's our signaling molecule or ligand. So that's triggering a response within the cell. So in this case, it's basically causing some sort of molecule to be activated, which is going to initiate the activation of, in this case, it's called a protein kinase. So this kinase 1 is now activated based on that relay molecule. And then that kinase, active protein kinase 1 will then activate another protein kinase, which we call 2. Okay, so you can see here we have a reaction take place with some cellular energy being used to activate this other protein kinase. And then that protein kinase 2 is going to activate an inactive protein. And that's going to initiate a cellular response. So we have basically a series of reactions that are taking place where we have one protein kinase is activated, which causes another to become activated, which causes another protein to be activated, until we end up having a cellular response. So that's what we call a phosphorylation cascade, because the protein kinases phosphorylates a protein. So you can see the little protein phosphorylation taking place there. Another example would be this, it's called the C-amp. So in the C-amp, this is an example of the G protein coupled receptor site that's going to be used. So remember that G protein coupled receptor, the GPCR, is going to receive the signal. In this case, it could be epinephrine. Okay, so that ligand is going to bind, and that's going to trigger the G protein to become activated. So that particular G protein has a source of energy called GTP. and that's going to activate an enzyme that's embedded in the cell membrane. So that enzyme, once it's triggered, once it's activated, it's going to basically cause this ATP to convert to something called CAMP, which is a type of messenger. And that's going to activate another protein to elicit a response. So we're still seeing the same pathway where we start with a reception, okay, with the G protein coupled receptor, and then we have a transduction. So that's going to cause the G protein to activate the enzyme. That's going to cause this reaction to take place to activate a protein. And then finally we get the response, the cellular response. Right? And then here's another one. So in this case, this could be an example of where we have a cell that needs to undergo growth, and so it's going to receive the ligand, which happens to be a growth factor, with the receptor site that's embedded in the cell membrane. And that's going to cause, so that's the reception part, and that's going to lead to this transduction pathway where we have the molecule, the growth factor, is going to trigger, in this case, a phosphorylation cascade, which will create eventually this activated protein kinase, which will enter the cell's nucleus and activate something called a transcription factor, which will initiate what's called transcription, basically where the gene gets activated and is going to be transcribed onto mRNA. And we'll talk more about that way in the future when we talk about protein synthesis. But same three steps are taking place. We have the reception, and then followed by transduction, and then now we have that elicit response taking place. So let's look at an example of the G protein coupling receptor.
Receptors that are plasma membrane proteins usually affect the cell through multi-step signal transduction pathways. These pathways allow for amplification of signals and signal coordination and regulation. Signal transduction often involves smaller molecules called second messengers and protein phosphorylation by enzymes called protein kinases. Second messengers are small non-protein molecules that act as intermediaries in signal transmission. Two important second messengers are cyclic AMP and calcium ions.
Cyclic AMP acts as a second messenger in pathways initiated by both G protein-linked receptors and receptor tyrosine kinases.
The hormone epinephrine acts via cyclic AMP as a second messenger. An epinephrine molecule docks with a receptor protein, which acts through a G protein to activate the enzyme adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP, or CAMP, which quickly diffuses through the cell and triggers further steps in the signaling pathway. In this cell, we show it activating a protein kinase. Calcium ions also act as second messengers in signal transduction pathways.
This time we will show a receptor tyrosine kinase, but G protein-linked receptors can also initiate release of calcium ions. The signal molecule docks with the receptor and activates an enzyme called phospholipase C. The enzyme cleaves a small molecule called inositol trisphosphate, IP3, from a certain kind of membrane phospholipid. IP3 docks with a calcium channel on the endoplasmic reticulum. The channel opens, releasing calcium ions into the cytoplasm, where they activate proteins that carry out cell responses.
Enzymes called protein kinases are important links in many signal transduction pathways. A protein kinase catalyzes the transfer of phosphate groups from ATP to another protein. More often than not, the activated protein is also a protein kinase, which may act on still another protein kinase. One kinase may activate many molecules of the next type of kinase in the chain, thus amplifying the signal until the last kinase activates many molecules of the protein that carries out the final cellular response.
Okay. So you can see, there's quite a bit of like complexity involved with cell signaling, but all of these examples come down to that three process pathway. So we always start with the receptor, and then we have transduction, and then we have a response. So, as far as the different types of receptors, we talked about that G protein coupled receptor, which involves the activation of a G protein, which will stimulate an enzyme to react. And in this case, that enzyme is converting ATP into that C-amp, or the CAMP, as they called it. And that will trigger another response to basically activate a protein kinase, which leads to this signal amplification process. But it will end in some sort of cellular response. So when we're talking about, the different receptors, those ligand-gated channels versus a G protein coupled receptor. Okay, so those you just need to know that's part of the reception phase. And then the different cascading effects that occur, like phosphorylation cascade, that's the transduction response pathways and so forth. So you can kind of just, glance at it in that regard. So focus on the reception, and then the transduction, and then finally the response. Okay. All right, so here's a question for you. What determines whether a cell responds to a hormone such as epinephrine? What determines how that cell responds? Okay, so what determines whether a cell responds to a hormone such as epinephrine? Well, that starts with that first phase, right? The reception phase. So it has to have a receptor for epinephrine, right? So if there's no receptor site, then there can we end right there. We can't have the reception phase take place. So receptor protein on or within that target cell will allow the cell to detect that signaling molecule, that ligand, that epinephrine, and that's going to cause a change. Okay, so if the cell responds, it's because there was that protein reception take place, that that epinephrine was able to bond to some sort of receptor site. Okay. All right, so that is all for chapter 5. Chapter 5. So for now, you can go ahead and finish up taking your notes with those course objectives. And then in the lecture exercise, if you're watching this before the lecture exercises, we're going to focus mainly on the phospholipid bilayer, the structure of the plasma membrane, as well as the different forms of diffusion and passive transport versus active transport. So, we'll focus on those, make sure that gets solidified in our brains. Okay, so I'll see you at the Zoom.